Okay, so chapter 6 is a tour of the cell. We're going to be talking about all the parts of the cell, all the organelles and components of both eukaryotic and prokaryotic cells. So the first thing we want to start out talking about here is cell theory. So this is a big overarching principle of components that have been shown to be true over and over again that is widely accepted. There are three components of the cell theory that we need to talk about. The first one is a cell is the smallest living thing. Now that we have already talked about back from chapter 1 where we did our organization chart and we said that a cell was the smallest unit of life. So that is the number one principle or component of cell theory. The other component of cell theory is that all cells come from other cells. So we're going to talk a lot about this when we get to chapters 12 and 13 and we talk about cell division. But that is the second component of cell theory, is that cells divide and multiply, and that's how we obtain new cells. The third one is one that we kind of talked about when we were talking about our characteristics of living things, and that this is that organisms are made of one or more cells. So we had a kind of a little debate about whether or not living things were cellular based, and from our view of an Earth perspective, at least so far of everything that we have classified as living here on Earth, we know that every living organism is made of at least one cell. So here, for us at least, we can say that life is cellular based, meaning that every living thing is made of cells. All right, there are three important features of cells that we need to talk about, and these are features of all cells. So it doesn't matter if they're prokaryotes, eukaryotes, plant cells, animal cells, doesn't matter what kind of cell it is, every single cell that we know of has these three features. Number one is the cell membrane. So every single cell has a boundary of a cell membrane that surrounds it. So if we look at this, in the picture here, the photomicrograph of cells, you can see this thin line surrounding all of these cells. Cell membranes control what comes into the cell and what leaves the cell, and every single cell has one. The second area that every single cell has is a DNA region. So in eukaryotic cells, we call this the nucleus. So that's in eukaryotes. In prokaryotic cells, which we know do not have a true nucleus, the DNA is found in the nucleoid. So in these cells shown in the picture here, we have a very well defined nucleus. And I'll show you what a nucleoid looks like in just a second. Now remember, the DNA is going to contain the instructions for making proteins. We'll talk more about that when we get to a later chapter of how that works. But DNA is like the owner's manual of the cell that tells the cell how to make all the proteins that it needs to make in order to do all of the functions that it needs to do. The last region that every cell has is called the cytoplasm, and this is basically everything in between. Anything that is not cell membrane and not nucleus, everything in between is referred to as the cytoplasm. So this is where all the organelles are. This is where a lot of the proteins are. All the other components of cells that are not DNA and not cell membrane are referred to as cytoplasm. And all living cells have these three features. So let's just refresh your memory real quickly about the difference between eukaryotic cells and prokaryotic cells. So remember, eukaryotes have a true nucleus. EU means true, and karyotic refers to a nucleus. So eukaryotic cells do contain a nucleus. They also contain what are called organelles. These are referred to as little organs. So these are things like the mitochondria, the chloroplast, the Golgi, the lysosome. So all of these we'll talk about as we go through the lecture today. So anything that fits into the domain eukarya, which we talked about back in chapter 1, so these are your protists, your fungi, your plants, and your animals, all of those contain eukaryotic cells. Your prokaryotic cell, remember the pro prefix means before, so these are things that would have evolved before nuclei. Prokaryotic cells do not contain a nucleus, nor do they contain other organelles. So in prokaryotic cells, remember the DNA is contained in that nucleoid. So it's a nucleoid region, not a nucleus. These are things that would fall into the domains bacteria and archaea. Okay, so what does that nucleoid region look like? So here's a prokaryotic cell, and you can see there isn't really a well-defined nucleus like we saw in the eukaryotic cells a couple of slides back. You can see all of these little squiggly lines in here where it's a little bit lighter. That is your DNA. That is the nucleoid region. So that's where the DNA is contained within the cell, but there's no well-defined nucleus. You also don't see any other membrane-bound organelles, so things like the mitochondria, the chloroplast, all the other types of organelles that we're going to look at. You also do not see those in a prokaryotic cell. And lastly, whereas in eukaryotic cells, when we talk about X-shaped chromosomes that sort of look like that, in prokaryotic cells, we typically see one big circular chromosome. Okay, so if we were to stretch this thing out and look at all these little wavy lines in here, we would see one large circular DNA, and that's characteristic of prokaryotic cells, different from eukaryotic cells. Okay, so let's talk about some of these organelles that we see in eukaryotic cells. So organelles, again, means little organs, and these are basically cellular compartments that are partitioned off by a membrane or surrounded by a separate membrane. They each perform a specialized function, and that allows us to sort of separate different activities within the cell. So for example, remember our condensation reactions? These are reactions that take little things and put them together to make a big thing. And then remember our hydrolysis reactions, where we take a big thing and we break it apart to make little things, okay? We would not want those types of reactions happening in the same place in the cell, because as soon as we make something, we might accidentally break it apart. So we would want to separate those two activities. We want to have condensation reactions on one side, hydrolysis reactions happening somewhere else. And that is a benefit or an advantage to having these organelles, is we can separate incompatible activities within the cell. Examples include the mitochondria, the chloroplasts are probably two that you're familiar with. We also have vesicles. That's another example we'll talk about in a minute. I refer to those as the UPS trucks of the cell. Their basic function is storage and transport of materials inside the cell. We'll talk about the Golgi. We'll talk about the endoplasmic reticulum. These are all examples of organelles. Okay, a couple of things we want to talk about the nucleus. By the way, the nucleus is an example of an organelle. It is a membrane-bound compartment that has a specialized function. The function of the nucleus, remember, is to hold the DNA or contain the DNA. So there are a couple of components of the nucleus that we want to talk about and be familiar with. The first of these is the nuclear envelope. This is the membrane. So it is a double membrane, so it has two layers of membrane, and it does have pores within this membrane that control what enters and what leaves the nucleus. Now, the nuclear membrane is actually even more picky about what it lets in and what it lets out than even the cell membrane, because remember, this is housing our DNA. This is our owner's manual for the cell. If something happens to that DNA, that cell dies. So we have to be very careful about what we let into that nucleus and what we let out of that nucleus. We don't want to have pores in our nucleus that allows our DNA to leak out. That would be very bad. We also don't want to allow enzymes inside that nucleus that might chew up or destroy our DNA. So we have to be very careful about what comes in and what comes out. And it is the job in the nuclear envelope to do that. We also, just like we have cytoplasm within a cell, we have nucleoplasm. So this is basically that semi-fluid interior part of the nucleus that is everything other than the chromosomes. So the chromosomes are the DNA, and then you have this sort of semi-fluid interior that is referred to as the nucleoplasm. We then have what's called the nucleolus. Nucleolus is contained within the nucleus, and these are construction sites for ribosomes. We're going to talk about ribosomes in a minute. The job of the ribosomes is to actually make proteins. So ribosomes make the proteins but it is the nucleolus that makes the ribosomes. So the ribosomes are made inside the nucleus and then shuttled out into the cell so that they can function in protein formation. So the ribosomes are made in the nucleolus, and then the ribosomes make proteins, or where proteins are made. We also then have our DNA inside the nucleus. So, and we have a couple of terms to describe that. So when we talk about a chromosome, we are talking about one DNA molecule with any proteins that might be associated with that molecule. But chromosome is singular. When we are talking about all the chromosomes inside that cell, we call that chromatin. Okay, and that is the plural form when we're talking about all the DNA and all of the associated proteins. All right, so let's look at this nuclear envelope a little bit more. So again, it is a double membrane system, so it has two lipid bilayers. So this is lipid bilayer number 1, and this is lipid bilayer number 2. Okay, so two lipid bilayers surrounding the nucleus. And surrounding the nucleoplasm. We also do have pores along that nuclear membrane that allows for exchange of material. So some things get to go in, some things get to come out, but again, it is tightly regulated. Okay, so now we want to talk about some of these organelles. And we specifically talk about the organelles that make up something called the endomembrane system. So this is a group of organelles that are responsible for making, modifying, and transporting material. So often these materials leave the cell and are used somewhere outside the cell, although they can stay internal and be used inside the cell. We are going to focus our efforts on making proteins. So your cells make other things as well, also lipids and some carbohydrates. But what the way we're going to talk about this and approach this today, we're going to be talking about the organelles that are responsible for making proteins. So there are a group of organelles that we're going to talk about. We'll start with the nuclear envelope, which we've already talked about. We'll talk about the endoplasmic reticulum. So proteins are made here. They then travel to the Golgi. They then travel through vesicles or lysosomes. We'll talk about a vacuole. Mostly this is going to be in plant cells. We'll talk about the vacuoles. And then we'll talk a little bit more about the plasma membrane, but we're going to spend a whole chapter on that in chapter 7. Okay, so let's start out talking about the ribosomes. Remember I told you the ribosomes are responsible for assembling proteins. So in this case, that is our sort of raw material. Sorry, let me move this. Okay, so that's our raw materials, and we're talking about assembling the proteins. So ribosomes we can have in two different places. So we can have out here what are called the free ribosomes. So they're not attached to anything, they're just floating out within the cell. Or we can have ribosomes that are bound to the ER. So these ribosomes are attached to what's called the ER. That is the endoplasmic reticulum. The ER is the endoplasmic reticulum. And when it has ribosomes attached to it, we call it the RER, or the rough endoplasmic reticulum. And that's because when we look at it under a microscope, the presence of those ribosomes kind of give it a rough or a textured-like appearance. Okay, so let's talk about the RER and what it does. There are actually two kinds of endoplasmic reticulums that extend off the nucleus. By the way, here's our nucleus right here. And so you can see right attached to that, we have the RER, or the rough endoplasmic reticulum, and we have the SER, which is the smooth endoplasmic reticulum. So RER is for rough, and this has ribosomes, or is ribosome positive, we'll say. The smooth ER, the SER, stands for smooth, and this does not have ribosomes. Okay, so the smooth endoplasmic reticulum does not have ribosomes attached to it, which is why it looks smooth under the microscope. Now, for our purposes, we're focusing on the RER, or the rough ER, because this is where proteins are made. The smooth ER is where we make lipids. Okay, so lipids are made primarily in the smooth ER, the SER. So we're going to be focusing our efforts again on protein production. So we're talking about the RER, the rough ER. This is where we take those polypeptide chains that constitute tertiary structure that we talked about a couple chapters ago, and we're going to put them together or modify them to make a final protein product. So let's talk a little bit more about the RER and the SER. So these are channel-like membranes. So you can kind of they look like little a maze, sort of, where you've got to find your way out, and they have these little channels and little pathways that the proteins or the lipids would work their way through. The key here is that proteins are modified, so we modify the proteins that are made by the ribosomes. Remember, the ribosomes make the raw material, the raw proteins, and it is here in the ER where we modify them. We specialize them. We give them their special touches that allow them to do their job. We make them into final proteins. From the rough ER, we're going to put them in a vesicle and ship them. Sometimes they can go directly to somewhere else inside the cell, or sometimes they go directly out of the cell. But for our purposes, we're going to continue down the endomembrane system. So we're going to put it in a vesicle and send it to the next organelle through this system. So remember, in the smooth ER, this is where lipids are made. So it's also responsible for destroying toxins inside the cell. But again, they don't have the ribosomes, so smooth ER is not involved in protein production. All right, so let's talk about the next organelle down the endomembrane system. So remember, we started here in the nucleus, we worked our way through the rough ER, and now we have packaged that protein up in a vesicle, and we're sending it to the Golgi. So here's the Golgi apparatus here. Again, it's sort of a stack of membranes, wash of membranes. Or a stack of membrane sacs is how we call it. The Golgi is going to receive and process proteins that arrive in vesicles from the ER. So we made our protein in the ribosome, we modified it in the ER, we put it into a vesicle, packaged it up in a vesicle, which again is kind of like the UPS truck of the cell, and we sent it to the Golgi. Now, if I were to give the Golgi a name, I would call this sort of the post office. Okay, so this is the UPS truck kind of going back to the post office to drop off our protein. And here in the Golgi, the proteins are going to be modified one more time and repackaged into new vesicles that allows us to ship them out of the cell. So this is the post office where we are packaging up these cells. We're giving them tags that we're either going to send them to the cell membrane, or we're going to send them totally out of the cell to a different cell. This is where they're going to get sort of sorted and sent on their way for where they need to go to do their job. The Golgi apparatus is also responsible for making lysosomes. So we're going to talk about lysosomes in a minute and talk about why those are important. So let's talk about that lysosome. If you see, the lysosome is sort of right here off of the Golgi. So here was the Golgi apparatus. The lysosome is right next to it. And basically, if you can think of it as Play-Doh, where you pinch off a piece of Play-Doh, that's sort of what happens to form the lysosome. We sort of pinch off a piece of the Golgi, and it becomes this separate little vesicle, and we call that the lysosome. So the lysosome is a vesicle that contains digestive enzymes. So this is basically the garbage truck of the cell. So this is anything that we don't need anymore, or if we made a mistake when we were making this protein and it's not what we needed to make, or it's not functioning right, old cell parts, any sort of bacteria, basically anything inside the cell that we need to get rid of is going to go into the lysosome. These digestive enzymes inside the lysosome is going to sort of chew up or destroy anything we put in there and get rid of it. It's the garbage truck of the cell. All right, so now I want to talk about a specialized organelle. So this is not necessarily part of the endomembrane system. But before I do that, I want to refresh your memory on the endomembrane system real quick. So remember, we started in the nucleus right here. That was kind of step one. We made it out then to the ribosomes, either the free ribosomes or the ones on the rough ER. So that's where we made our raw material proteins. We then, we made our raw material proteins in the ribosomes. Then we sent that protein through this sort of windy maze of the ER where it was modified. It was then packaged in a vesicle and sent to the Golgi. Here, let me do that in a different color. So here's the Golgi. That's where it was repackaged and shipped either out of the cell or into the cell, wherever it needed to do its job. Most of the time, I'll say out of the cell. But we kind of put an address on it, we put a tag on it, we sorted it, and sent it on where it needed to go. So that is the endomembrane system from kind of start to finish. We start in the nucleus with the instructions. We go to the ribosomes where we make the raw material. We go to the ER where we modify it or specialize it. And then we send it to the Golgi where it gets repackaged and shipped out of the cell. And then lastly, of course, it hits the plasma membrane, which is the gateway to send that protein out of the cell. So that is the endomembrane system. Now I want to talk about the mitochondria and sort of the specialized function that the mitochondria holds in the cell. So the mitochondria, you might have heard it referred to as the powerhouse, right? If I had to give this a designation, I would call it the power plant of the cell. This is what drives every process that the cell does. It is involved in production of ATP, which is the cell's energy currency. Without the mitochondria, we can't produce ATP. We would not have the energy that we need to carry out all the cellular processes that we have to do. This is also, just like the nucleus, a double membrane system. So there are two lipid bilayers that surround the mitochondria. So there is an outer membrane out here, and then there is an inner membrane in here. We're going to talk way more about this membrane system when we get to cellular respiration. You might know that mitochondria have their own DNA separate from nuclear DNA. Mitochondria has their own DNA. We refer to it as mtDNA. It is typically passed down through the mother only. So your mitochondrial DNA is passed from your maternal side. Mitochondria divide on their own, so they have their own division process separate from the cellular division. They also have their own ribosomes, so they can carry out their own protein production separate from that of the rest of the cell. And again, it's referred to as the powerhouse of the cell because it provides the energy for everything that cell does. Okay, so now I want to talk about a couple of organelles that are specific to plant cells. So the first of these that I want to talk about are the chloroplast. You guys probably remember talking about the chloroplast in some of your biology high school classes. So this is the organelle that is important in photosynthesis. So again, we're going to talk a lot more about this. We're going to spend a whole chapter on photosynthesis. But this is the organelle that is responsible for carrying out this process and contains the chloroplast. So the chloroplast within the chloroplast contains the chlorophyll. That's the pigment that is responsible for carrying out photosynthesis. So it carries out the reaction of sunlight plus CO2 yields glucose and ATP. So these are the food sources for the cell. This is how plant cells make their own food. And again, we'll talk much more about this in a later chapter. So chloroplasts are found in photosynthetic eukaryotes, which are predominantly plants. There are some bacteria that carry out photosynthesis as well, but again, bacteria don't necessarily have the same chloroplast structure that we see in plants. There are two membranes. So again, just like in the mitochondria, we see this double membrane system with an outer membrane and then an inner membrane. If we look here, we have the two membranes, and we'll talk again more about that when we go through the process of photosynthesis. There are two parts to the chloroplast which are going to be important. So one is called the thylakoid. So that's these little stacks that look like stacks of pancakes. We refer to those as the thylakoid. Thylakoids. And there's a part of photosynthesis that's carried out in the thylakoids. And then there is this sort of semi-fluid interior in the inside, and that's referred to as the stroma. So that's the sort of semi-fluid interior, just like we had chloroplast and nucleoblast. It's sort of like that, but it's referred to as the stroma in the chloroplast. So again, these also contain the photosynthetic pigments, which the most popular one is the chlorophyll. We'll talk about others when we talk about the photosynthesis chapter. But all of that is contained in the chloroplast, and that's what allows the plant to carry out photosynthesis. The last organelle that I want to talk about, again specific to the plants, is the central vacuole. So this is a large membrane-bound sac. It is only found in plant cells, so plant cells only. It contains or stores water, amino acids, sugars, ions, and other waste products for the cell. It's a large membrane sac, and it actually composes about 50 to 90% of a plant cell is this vacuole. So mostly there is water inside this vacuole. So you can kind of think about it, if a plant cell has been watered, so if you've watered your plant lately, if you have, your little pot and you have a little plant here in your pot, if you water it and provide water to those cells, so here is my plant cell, and basically here is my central vacuole. I'll call it my CV. So if that CV is full of water, because we have just watered our plant, it's going to be big and full and pushing against the sides of that cell wall. And so we're going to have a very rigid plant cell that is happy and healthy, and it's going to make our plant stand up nice and tall. However, if we have a plant that we haven't watered lately, right, our plant is going to start to droop. That's because, so here's our plant cell, that's because our CV now looks like this. It's much smaller. There's not enough water to fill it up and make it push against the outside of the cell. And so the cell itself is going to start drooping a little bit. And when the cells start to droop, the plant starts to droop. So you can see the role of a central vacuole in keeping our plants nice and healthy and tall and standing up straight. All right, so that is the end of chapter 6. I will see you in class.